our past classes, we've been discussing prokaryotic cells, the archaeobacteria, and the true bacteria. We suggested that as a group, their extreme diversity of habitat and their diversity of metabolism is evidence that they've been around the planet for a very long period of time. In addition to these inferences, there's evidence from fossil record that suggests that the earliest living things on the planet were similar to modern bacteria in many ways. Furthermore, when we compare the structure of prokaryotes to eukaryotes, we see a simpler structure. We can presume that the simpler structure arose first. So as we move into the domain eukarya, as we move into the domain eukarya, discussing the kingdoms Protista, fungus, plants, and animals, the first question we need to address is, how did the eukaryotic cell structure come into being? In other words, what's the origin of the nucleus, and how do we understand the presence of large membrane-bound organelles such as the mitochondria and the chloroplast? The best hypothesis we have for the origin of the nucleus is that it arose from an infolding of the cellular membrane. This drawing shows what biologists think may have happened. The outer membrane folded in, over time, surrounding the genetic material, encasing it in a nucleus. This compartmentalization of the nuclear material, separating it from the rest of the cell, became an advantageous structure, giving us a nucleus. Now, the suggestion for the origin of the mitochondria and the chloroplast is a hypothesis, or is based on the concept of endosymbiosis. So let's talk about endosymbiosis. Is the suggestion that the organelles were at one point their own independent cell. So we have two bacteria. The larger bacteria engulfs the smaller bacteria, but instead of digesting it, the smaller bacteria lives inside the bigger bacteria in a mutualistically beneficial relationship. Over many generations of this relationship passed along, the smaller cell started to lose some of its cellular components and evolved into an organelle. We have evidence to support that the origin of the mitochondria and the origin of the chloroplast came about through endosymbiosis. Let's look at some of that evidence. The endosymbiote hypothesis was first proposed by Lynn Margulis in 1967. Her hypothesis originally proposed that the mitochondria are the result of the endocytosis of an aerobic bacteria, and that chloroplasts were the result of the endocytosis of a photosynthetic bacteria. In both cases, a larger anaerobic bacteria that would struggle to exist in an ever-increasing aerobic environment was the cell doing the engulfing. This arrangement became a mutually beneficial relationship for both cells, or a symbiotic relationship. So here we have a cell engulfing an aerobic bacteria that over time evolved into mitochondria. And down here we see one of these cells that's already incorporated a mitochondria engulfing a photosynthetic bacteria that over time evolved into a chloroplast. We can also see in this diagram not a cell lacking a nucleus, and over time infoldings of the outer membrane, creating a nuclear membrane around the nuclear material, around the DNA. Now, another piece of evidence that supports this hypothesis is when we look at the structure of the mitochondria, for example. There's distinctly two separate membranes here, an outer membrane and an inner membrane. If a bacteria is engulfed by another bacteria, the engulfing process would wrap the bacteria in a membrane package, and it would have its original membrane on the inside, and so that this double membrane structure is explained by this process of engulfing, or endocytosis. Other evidence that supports the endosymbiote hypothesis, besides the dual membranes, is the presence of DNA in the organelles, so that mitochondria and chloroplasts have their own small pieces of DNA, which is unusual. Not only do they have the DNA, but it's single and circular, like bacterial DNA. The organelles will divide within the cell, and that division looks a lot like the process of binary fission, or bacterial reproduction. These organelles have their own small ribosomes that are similar to bacterial ribosomes in structure. And when we look at the size of modern-day mitochondria and modern-day chloroplasts, they're similar in size to bacteria. All of this supports the endosymbiont hypothesis, which is now known as the endosymbiont theory. So if we start with the simple structured bacteria, and we look at the complex structure of a modern-day eukaryote, the two big things that we have to kind of account for is the presence of the nucleus and the large membrane-bound organelles. We have suggestions as to how each of these has come about. So we can move on to the domain eukarya and the kingdoms that are inside of it, including the protist, the fungus, the plants, and the animals. And in each case, for the fungus, plants, and animals, we have a very clear-cut definition. It's very easy to kind of distinguish what a fungus is compared to a plant, compared to an animal, based on a simple set of criteria in terms of looking at the number of cells, heterotrophic versus autotrophic, cell wall or no cell wall. The trouble comes when we try to distinguish the protists. They don't fit as nicely into a box. So our first subunit within the eukarya, the first subset group that we're going to look at, are the protists. Well, that finishes our introduction to the kingdom eukarya and the origin of the eukaryotic cells. The next video will cover protists and the different types of protists and the characteristics we see there.